And remember, the innate immune response and the acquired immune response starts at the same time. They start at the same time. One will protect you while the other one is being activated. And one of the things that I don't think people understand is they don't understand that this dendritic cell, say it's a Langerhans cell, or it's, let's go ahead and make this full view. This dendritic cell is going to be activated in the tissue, in the skin, in the body someplace by PRRs on its surface. Or if it's a macrophage, it's going to be activated by PRRs on its surface. If it's a B cell, what activates a B cell has to be signal 1. When we talk about a B cell as an antigen-presenting cell, it's different from macrophages and dendritic cells. Signal 1 that allows a B cell to become an APC is cross-linking of 2 B cell receptors on the surface of 1 B cell. And the reason that is if you think about the lymphatic system, we're going to talk about the lymphatic system. When an organism drains into the lymphatic system, if PRRs on any B cell could activate any B cell, then all B cells would become activated, correct? And we don't want all B cells to become activated. So only those that have been cross-linked by the pathogenic epitopes, only those B cells will be activated. Does that make sense? Okay, so we're going to activate this dendritic cell, and it is innate. If it's just a regular old macrophage, if it hasn't had any PRRs activated, it's just a regular old macrophage, a Kupffer cell, a mesangial macrophage, a macrophage in the blood. A Langerhans cell is immature in the skin. It will only become an APC once its PRRs are activated by MAMPs or DAMPs. Are we good? Okay. Then this dendritic cell here is going to hop into the lymphatic system. Okay? Before we talk about that, we're going to go back over how the T cells get activated and what is positive and negative selection and what happens in positive and negative selection. So if you look at this part, that's positive and negative selection. And that is where the T cell receptors and the B cell receptors get their receptors. Their B cells and T cells get their receptors. That's where your VDJ recombination or your VJ recombination for your light chain happens in the primary lymphatic vessels, lymphatic organs. So name a primary lymphatic organ, anybody. So bone marrow. What's the other one? Primary lymphatic organs. That's where these, so if you go back to the hematopoietic stem cell, that hematopoietic stem cell is going to generate granulocytes, it's going to generate monocytes, it's going to generate pre-lymphatic cells, pre-T cells, pre-B cells. They're going to migrate to their primary lymphatic organs, and they're going to get their T cell receptors. And if their T cell receptors don't work, they're going to be killed. If they can't bind to MHC molecule, they're killed in positive selection. If they bind too strongly to self-peptides, meaning that they would recognize a self-peptide, we're going to kill them in negative selection. T cells. B cells will also undergo positive and negative selection no matter what the book says. It's very similar in that if the B cell receptor can't send a signal, they'll be deleted. If the B cell receptor sends too strong a signal when it binds to self in the bone marrow, it will be deleted. Okay? Now we're going to talk about one exception to the negative, the two binding too strongly selection in just a minute. But generally, that's the way we get our B cells and T cells. And then once they have been generated, once they can send a signal, once they recognize your MHC molecule, then what they're going to do is they're going to go on and migrate to the secondary lymphoid organs. Okay? And here, as you can see here, this is the primary lymphoid organs, and now they talk about this is the secondary lymphoid organs. So they have now, once they have become an immature, or they've gone from an immature cell to a mature naive B cell or my mature naive T cell, they're going to migrate to your Skin lymphoid organs, your salt, your skin-associated lymphoid tissues, or your mucosal-associated lymphoid tissue, your malts. And depending on, they're like, well, how do they know which one to go to? They circulate all the time. I just went to a lecture where they talk about how these circulate from lymph node to lymph node, from place to place. There's a very complicated set of regulated chemokines that allow naive T cells and B cells to migrate from place to place. You guys don't need to know how that works. Magic. If you want to learn how that works, come with me to France, okay? We'll talk about it in my immunology class then. That's above your pay scale in this class. Are we good? Okay. So we are going to go to the lymphatic tissue. And when we talk about the lymphatic tissue is different than the circulatory system. It is open-ended, meaning that this is a lymphatic vessel. And notice it's not a closed system like your circulatory system is. Your red blood cells stay inside your red blood cells. They do. Your white blood cells stay inside your white blood cells unless they're called into the tissue someplace. Somebody asked me, well, I don't understand the difference between an extracellular bacterium and an intracellular bacterium when we're talking about an antibody response. So when we're talking about an antibody response or a bacterial infection in the tissues, what I want you to realize is that unless it's living inside the cell, an extracellular bacterial infection will live outside your cells, in between your tissue cells. They'll live in your muscles, they won't live inside the cells, they'll live in your muscle tissue. But your cellular plasma membranes will keep those bacteria outside of the cell. And if they're outside of the cell, things like complement, things like antibodies, those can all act on an extracellular bacterial infection. Okay? Also, as long as a virus is outside of the cell, we can actually make a neutralizing antibody response that's going to stop that virus from being able to bind to a cell and get inside into the cell. Once something is inside the cell, the only way that we can eliminate it is by killing the cell off by a T cytotoxic cell or something called an NK cell. We're going to talk about it.
Are PRRs intracellular or extracellular?
Our PRRs, it depends on the PRR. Name intracellular PRRs. NLRs and RLRs. What would be extracellular PRRs? TLRs on the surface will bind to anything that's outside of the cell, especially macrophages and dendritic cells. If they phagocytose or endocytose those, as they bring them in and those things are destroyed or acidified, then that DNA of viruses, or that DNA of bacteria, or that DNA is exposed in those endosomes, and then those PRRs are activated, those TLRs that are in the endosome become activated to that DNA, or that RNA. Does that make sense? Okay, so when we talk about those, depending on where they are, remember that every cell in the body can make interferon alpha and beta, which is why every cell in the body makes RLRs, because RLRs bind to viral RNA. We did talk about this before. I want you to know that it's true that all viruses will induce some interferon, alpha and beta. Even if it's a DNA virus. Herpes is a DNA virus. It induces interferon, alpha and beta, but worse than influenza. So there are some that produce a very strong interferon alpha and beta response. There are some that produce a weak interferon alpha and beta response, but we generally always see an interferon alpha and beta response eventually. There are some viruses that are really good at being sneaky and specifically doing things like send off an interferon alpha and beta receptor into your serum, into your body fluids, so that that interferon alpha and beta can't bind to neighboring cells to warn them. It's soaked up by those floaty viral proteins and it's taken away. So we can't alert the body as well that there's a virus there. So there are some viruses that are sneakier than others. Generally, the ones that cause more severe disease are the ones that are sneakier. Obviously, the common cold doesn't have any of these abilities because we can eliminate it pretty quickly with just the innate immune response and some IgA. Okay? Does that make sense? So this is the lymphatic vessels. You guys need to know the difference between a lymph node and a Peyer's patch. Where are Peyer's patches located? In the intestine, in the gut. So when we're looking at all of these things here, when we're looking at all of these things in this person's gut, most of those are Peyer's patches, and most of those surround the large intestine because the majority of all the bacteria in your body are in your gut, in the tube of your oral cavity through your anus. That's where the majority of them are. We have some that live on our skin, we have some that live in our nose, we have some that live, they found them in our respiratory tract. There are normal commensals everywhere, but where we find the biggest, most absolute amounts is in the gut. So we're going to need a lot of sentinels to make sure that those organisms that are in the gut stay in the gut, or that our normal microbiota are safe and good. So what we do is we have these lymphatic vessels. So for a lymph node, the fluid comes in. So again, this capillary, the fluid leaks out of the blood. This is plasma in the blood. It is interstitial fluid in the tissues, and then when it becomes drained into your lymphatic vessel, it becomes lymph. It's just the same fluid, different locations. Okay? You guys know what happens. One of the things, we have a heart that pumps the circulatory system, and just like we talked about with sepsis, if there's not blood pressure that allows the heart to pump blood effectively, the heart starts beating really rapidly because blood pressure has dropped. There is no pump for your lymphatic vessels. There's no pump. It is the movement of your muscles. As you move your muscles, as you move, that forces that interstitial fluid either back into the capillaries or forces it into a lymphatic vessel where it becomes lymph. You guys know, you've seen all these places, or if you've ever been in an old folks home where a nurse is taking someone's leg and moving their leg to try to keep them from getting, because they're just laying in one position, they're not moving their muscles, they get fluid buildup and they have a lot of fluid in their body. So what you're doing by simply moving the leg is forcing those muscles to work to push that fluid into the tract and get it back into the bloodstream. Does that make sense? The same thing is true if you're walking a 5K, like me, and you're not very good at exercising, and you forget to move your arms very much, you all of a sudden see that your hands are bent, they look like a meat mitt, right? And then all of it's because they're full of fluid. Because you haven't been moving your hands, you've been moving your legs. So your heart has been pumping, you're getting a lot of fluid in your tissues. And what do you do when your hands start to fill up with fluid? Somebody told me, yeah. Everybody, what do? You're moving your muscles to force that fluid into your lymphatic vessel or back into the capillaries. So muscles are what drive the fluid motion into the lymphatic vessel. And one of the most important things about the lymphatic vessels are, if there is a bite in the arm, you can understand that the bacteria or the pathogens will drain into the nearest lymphatic vessel, percolate around the B cell follicles, percolate through the T cells. Now, T cells won't react at all to things percolating through the paracortex, but if there's a macrophage or dendritic cell in the paracortex, those will react to those pathogens, become activated by PRRs, start activating the T cells in the lymph node to become activated so that they can go on to help the B cells. Does that make sense? Okay, so we have the ability, not only that, but if it's a Langerhans cell, say you're a Langerhans cell, you become activated in the skin, you have to migrate from wherever this cut happened into the nearest lymphatic vessel. You're going to use that lymphatic vessel as a highway to get to where you need to go, so that you can get into the paracortex region and find naive T cells. Naive T cells will come and test you to find out which T cells are going to be activated and which T cells are going to undergo clonal expansion. Right? How many T cells are going to do that? Very few. Everybody's going to come along, look at the MHC on that dendritic cell, and then they're going to go off and wait until their pathogen is encountered. It has to be on MHC molecules, so on the way from that skin, the dendritic cell has upregulated B7. it has downregulated self-peptides, and it is loaded pathogenic peptides into its MHC class 1 and class 2. 8 to 10 amino acids in its class 1, 25, 13 to 25 for its class 2. So there's going to be different sections of that pathogen presented in different molecules, but that's fine. It's from the same, it's from measles, if you have a measles infection. It's from a cold, if you have a cold. It's from syphilis, if you had a bad encounter last night, okay? Halloween is bad, okay? Lots of tall, dark figures. So, one of the things you need to do is you need to make sure that the dendritic cell gets activated and migrates via the lymphatic vessel to the nearest lymph node, because that's where the T cells are. Somebody always said, why did Willie Sutton, there was an old bank robber named Willie Sutton, and he robbed banks. And they said, Willie, why do you rob banks? And he said, because that's where the money is. Why do dendritic cells migrate to the lymph nodes? That's where the naive T cells are that I need to activate. Not the follicles, the T cell region, because antigen-presenting cells are going to activate T cells. Okay? So, Peyer's patches are different. They have to always, you can see this dendritic cell here, and I'm going to have a figure on this, I'm going to ask you about Peyer's patches on the exam. Peyer's patches are very important. They have a number of different features that are different from most lymph nodes. They have these dendritic cells that reach out their dendrites to check to make sure that what is normal microbiota is normal. Because remember, as you're growing up, as you're going from birth to childhood, your immune system is going to, your bacterial microbiota is going to mature into an adult microbiota. We're going to see what normal is. Your dendritic cells know what normal is. They're going to go up and they're going to take like a periscope up through your mucosal tissue, take a sample of what's out there and check to see if everything is okay. If things have changed, they will be alerted. And there's ways that you guys don't have to know how these things know what's normal and what's not. They can detect these changes. There are all sorts of really complicated methods that we can detect changes. When you get a salmonella infection, you're not supposed to have salmonella. It's a pathogen. E. coli, normal. If it's O157H7, not normal. Okay? All of these things can happen. So we're going to have these dendritic cells that reach through and sample the gut microbiota. We are also going to bring things in through these M cells. So we're going to be constantly bringing stuff in through the M cells to make sure that what is normal and what is not normal. Some pathogens use that as a mechanism to get in. Does anybody remember a pathogen that uses the M cells to get in? Shigella. So Shigella comes through the M cells and can infect you by, they can trick out the system where they can get in through the M cells. So we have this ability to get in through the M cells, and then it causes then those macrophages get infected by Shigella. The Shigella grows in the macrophages, and it reinfects the mucosal epithelium. And the way that Shigella stonii causes disease is it migrates from cell to cell by the actin tail that is created in that cell, and it causes an inflammatory response in that single layer of mucosal epithelium that's in your gut, and it causes them to have inflammation, they slough off, you have diarrhea because your body is trying to flush away those organisms. Innate immune response, activating diarrhea or vomiting to try and get rid of this organism that's trying to infect more of your gut mucosal tissue. So diarrhea is an innate immune response. We have Peyer's patches. The Peyer's patch is the name of that whole tissue. The area where the T cells gather is, we're just going to call it the paracortex, similar to what's in the lymph node, because the follicles are still B cell follicles, right? So we're still going to call the B cell follicles B cell follicles. We're going to call the lymphatic vessels that bring the lymph into the lymph node are called afferent lymphatic vessels. I like to think of them as A first. They bring the lymph into the lymphatic vessels, into the lymph nodes. That lymphatic fluid percolates throughout that lymph node or Peyer's patches, and then it drains out of the efferent or exit, efferent lymphatic vessel, and if it hasn't been activated or if there's a lot of pathogens there, it will flow onto the next lymph lymph node, or you will activate more T cells and B cells. And just trust me that the right T cells and B cells become activated. But that's why it might take a day or two for you to get an immune response for those organisms to get to the B cells, because only a few B cells can respond to any one given infectious organism. So we have to find those B cells, activate them, cross-link them, allow them to migrate to the edge of the B cell follicle, and wait for help. T cells have to wait for that antigen-presenting cell to get to the paracortex to activate them. It can take a day for that to happen. And then they have to find them, and they have to undergo signaling. And signaling can take hours. It has to take hours to make sure that those T cells are the right T cells. Because if those T cells aren't the right T cells, then we can have an autoimmune response, where we activate the wrong T cells and they start targeting your cell tissue. That can't happen. So this is why the acquired immune response, we're going to activate a handful of T cells, a handful of B cells, because there are different epitopes on the surface of every organism. The T cells can recognize something completely different from the B cells that get activated to the same ones, because the T cells have to recognize a linear peptide in an MHC molecule, not a B cell which can recognize a lipid or a carbohydrate or a protein epitope on the surface that's not processed in any way. So a globular protein can activate a B cell. A T cell has to be activated by a linear peptide in an MHC molecule. Does that make sense? Okay, so we talked about MHC molecules, we talked about this before. T cells are MHC restricted. So we have the dendritic cell migrating into the paracortex, we're going to activate T cells. So when we activate T cells, notice the antigen in this overview from the book. The dendritic cell, which is the primary, the best antigen-presenting cell in the body, its job is only to be an antigen-presenting cell. That's what a dendritic cell is supposed to do. A macrophage is a garbage man until it gets activated by PRRs to be an antigen-presenting cell. Okay? I like to think of him as David Banner, right? Once he gets activated and angry, he becomes an activated antigen-presenting cell. He can become a superhero.
I just want to ask, so to what extent is this predicated on some cells already having phagocytized and destroyed some of the pathogen? Like if you have an encapsulated bacteria that's good at avoiding phagocytosis, is it harder for dendritic cells to find antigens in the extracellular fluid to grab?
So one of the things that happens, that's a great question. So one of the things, the reason why some organisms have pathogens is because remember that movie I showed? That movie where they're chasing around, it takes longer for them to get phagocytosed and present and get to a lymph node. They can outwit the immune response for a short period of time. But again, one of the organisms that we know that does that is Strep pneumoniae. Strep pneumoniae has over 90 different types of capsules. We make a conjugated vaccine and a polysaccharide vaccine to all of those to some of those capsules, the most common disease-causing capsules. We're going to talk about what a conjugated vaccine and a polysaccharide vaccine again in just a minute. Okay? But we have a long way to go, so I'm going to get going. Notice that, again, how long it takes for us, how long the incubation time is, how long it takes for a dendritic cell to become activated, how long it takes for a macrophage to find and become activated. It's going to be different for every organism because every organism has different incubation periods. Every organism has different prodromal. Every incubation time has an acute phase that's different. Every organism is going to be different and have different ways of trying to establish an infection in your body. So there is not one, it's going to take a day. Sometimes it takes longer, sometimes it takes shorter. It depends on the pathogen.
Would there be a question on the test, like this organism has an incubation period of 2 days, how long would it take for
No. She asked, is there going to be a question on the test that this organism has an incubation phase of 2 days, how long is it? No. Do you have to know the incubation period anyways, unless it's short or long? Rabies, long. Cold, short. Influenza, short. We good? So, Notice that the dendritic cell is coming into the secondary lymphoid organ, Peyer's patch, a lymph node, via the afferent lymphatic vessel, if it's a lymph node. They're going to come in, they're going to find T cells migrating into the T cell region and activate naive mature T cytotoxic cells or naive mature T helper cells, depending on what the immune response needs. The immune response knows what it needs because those PRRs that were activated indicates to the immune response that this is an extracellular bacterial infection, or that this is a viral infection, or that this is an intracellular pathogen. It has the ability of, because those PRRs have been bound on that antigen-presenting cell, that antigen-presenting cell knows what to tell the T cells that we need. Do we need to make both a T helper cell and a T cytotoxic cell response? It depends on the pathogen. There are some pathogens in which you don't need a T helper cell response, you only need a T cytotoxic cell response. And you don't need a B cell response, only a T cytotoxic cell response. There are some pathogens where you need both a T helper cell response and a T... Did my mic just go off? Oh, of course. There are some pathogens okay, there's got to be batteries. Okay, so there are some pathogens that need both a T helper cell response, and there are some pathogens that need a T helper cell response, an antibody response, a T cytotoxic cell response, you name it. And I can't get this open, of course. Okay, so what are we going to do? So again, there is no one best answer. However, if you're a virus, you're going to need a T cytotoxic cell response to help stop. If you're a virus, you need a T cytotoxic cell response to stop the infection. Absolutely. You're going to need, if you're creating a vaccine, you're going to want to make an antibody response that neutralizes that virus before it ever gets into the cell, so that we're going to have both. A vaccine could prevent you from needing a T cytotoxic cell response. Some vaccines actually specifically generate a T cytotoxic cell response to help protect you, so that you have memory cells at the location this organism is going to get in, so that as soon as the organism gets in an infection, those T cytotoxic cells will only need signal 1 to become activated and kill that cell. Boom, kill. Before it can undergo a lot of replication, before it can infect new cells. Does that make sense? Okay, so let's talk about this. Dendritic cells activate T cells. Notice that that B cell is activated by an activated T helper cell. B cells are not activated by antigen-presenting cells. B cells can be antigen-presenting cells. So what else do we know about this? This is exactly the same thing. This is another figure from the book showing the same thing. Notice the dendritic cells coming into the T cell region, activating naive mature T cytotoxic cells or naive mature T helper cells. They clonally expand once they're activated, and once they're activated, they go on to do the things that they're supposed to do. Once T helper cells get activated, all T helper cells will upregulate something called CD40 ligand on them to help activate a B cell or a macrophage. It can help make macrophages stronger, faster, angrier. We'll talk about this in just a minute. Notice that it is the T helper cell that is going to come up here and help this B cell. The B cell, remember, the T cytotoxic cell and the T helper cell need 2 signals at the same time. This B cell has been cross-linked when this dendritic cell got activated. It's going to clonally expand a little, make a little IgM, go to the edge of the B cell follicle and call for help. So let's talk about that now. This is a question from Pull Everywhere. Let's get Pull Everywhere started. Okay, what happens when a mature naive T helper cell receives only signal 1? What happens when it receives only signal 1? Go, talk amongst yourselves. You have a minute. Go. Talk with each other. It's only receiving signal 1. This is a mature, naive T cell. What does it have to have in order to become activated? What happens if it doesn't receive that thing? Talk to Alec. Anybody who has questions, raise your hand. Alec is here. So is Haley. Haley is here. Walk around, ask questions. You have a minute. You have 30 seconds. Seconds. Okay, 5 seconds. 4, 3, 2. Is everybody in? Okay. It is deleted or energized. If a naive T cell, that could be true for a T cytotoxic cell, a naive T cytotoxic cell, that is true for a naive helper cell. A naive helper cell is a TH0, it's a T helper cell 0. It doesn't know what kind of T helper cell it's going to be until it's told what to be by an antigen presenting cell. A T cytotoxic cell is going to kill. It has to be licensed to kill, though, by 2 signals. We're good? Okay. Let's talk about T cytotoxic cells and T helper cells. Question? Oh. We're going to talk about the difference between signal 1 and signal 2 for the next 45 minutes. Okay? So, we're going to talk about what is signal 1 for a T helper cell. What would signal 1 be for a T helper cell? MHC class. For a T helper cell. For a T helper cell is 2 with a pathogenic peptide on it that has been uploaded on the way to the lymphatic vessel. For a T cytotoxic cell, signal 1 is what? MHC class 1. What is the co-receptor in a T helper cell? Oh dear God. Okay, it should say, I think, it doesn't say here. Oh yes, it does. What is a co-receptor in a T helper cell? CD4. What is a co-receptor in a T cytotoxic cell? What is a co-receptor associated with? It's the co-receptor. It's associated with the, say loud, with the receptor, the T cell receptor. So that is part of signal 1, correct? Signal 2 is going to be B7 that was upregulated. B7 is not on an antigen-presenting cell until, it's not on an immature antigen-presenting cell. It is not on a Langerhans cell in your skin. It is not on a macrophage. It's just eating normal cell proteins. It's not on anything. It's not on a B cell. B7 is not present until activation has occurred for the antigen-presenting cell. For B cells as an antigen-presenting cell, that will be cross-linking. For dendritic cells and macrophages, that is activation by PRRs. Then, once it gets activated, on the way there, it is going to downregulate cell peptides, upregulate. Does everybody know what downregulate and upregulate means? It means we're going to take it off the surface of the cell and we're going to put it up. I want everybody who's born today to raise your hand. No one is born today. Okay? Everybody who's born in November, raise your hand. Thank you. Okay, let's go. Raise your hand. Okay, you will raise your hand. Raise your hand. Who's born in November? Raise your hand. I'm not going to force you to do anything stupid. I promise. Just sit there. These people have upregulated a receptor, so they would be the dendritic cells. Keep raising your hands. They have upregulated B7 on the surface of their cell. No one else here has. You have been activated by PRRs. You've upregulated B7. You can put your arms down. Okay? Does that make sense? So on the way to the lymph node, they have become a professional antigen presenting cell. They're going to upregulate B7 on the way there. All naive T cells will have a T cell receptor and a co-receptor, depending on what flavor you are. You will also always express the CD28 molecule so that you can be activated anytime there's a B7 in your presence. All T cells express CD28. Only activated antigen-presenting cells present B7. We're going to come into the B cell, the paracortex, we're going to activate you by giving you signal 1, which is MHC and peptide binding to your receptor and your co-receptor, and then giving you signal 2, if you recognize that allele and that peptide is your pathogen that you are responding to, then you will also bind to the B7, but only if your T cell receptor recognizes that peptide. If it doesn't, you test that peptide and you walk away. Your job is to walk away if it doesn't fit your T cell receptor's antigen binding site. Right? So, you have to get the signals at the same time because we are making James Bond. He's going to be licensed to kill it for your T cytotoxic cell receptor or a T cytotoxic cell. We can't just let a bunch of James Bonds, you know, that would be interesting, run amok in the world, right? We only want to activate him when there's a specific threat to the body. Okay? So, we're going to activate that. Signal 1 is MHC and a pathogenic peptide. Okay? Binding to the T cell receptor and the co-receptor, depending on, and this is from this book. If it gets 2 signals, then the T cells proliferate and differentiate. If it only gets signal 1, then it dies or undergoes energy. This figure from the book explains that. So if you don't know, print that off, put it on a whiteboard, and explain this to yourself. Okay? So what else do we know? We know that when I go into an actual immunology review, this is what they show me. In order to get signal 1 and signal 2, this is the antigen-presenting cell on the right. This is the T cell on the left. Is it a T helper cell or a T cytotoxic cell? It is neither, because it's not a CD4 or it's not a CD8. It's both. So it's showing you that that receptor is going to bind to whatever that MHC molecule is. Notice it's not involved in antigen recognition. It's just involved in helping stabilize that interaction. That co-receptor is actually vital in activation. It sends a signaling molecule, helps activate the signaling cascade. Without that receptor, the signaling cascade is not activated. So we need CD4 or CD8 to bind to the MHC molecule, while the T cell receptor, this unique specific T cell receptor, and notice that T cell receptor is recognizing both the MHC allele and the peptide. That specific shape. That's an allele, that's one of the, if it's MHC class 1, it's one of 6 of the alleles. It's a specific allele shape and a specific peptide that's presented in that allele that it recognizes. Your alleles, because it's tested on your MHC molecules in your body. Are we good? So, we're going to activate this T cytotoxic cell to become, or this T helper cell. You guys do not need to know any of those other molecules. In France, okay? Here you need to know the 4 that I talked about. You good? Okay. CD28 is a molecule that's on the T cytotoxic cell. It helps also send a stabilizing signal. Again, bigger than past your pay grade. Just know that the co-receptor and CD28 are vital in sending a signal into the cell that says you have been activated. If it only receives 1 signal, it sends a signal, you must die now. Or become energized. It has to happen at the same time. B cells get cross-linked and wait. Because they have to wait because we have to activate the T helper cell. So it makes sense that they're going to get activated, make a little IgM. IgM. Okay? We're going to make a little IgM and wait to be told what better antibody to make. By an activated effector T helper cell. We good? So when we talk about this, what is the second signal necessary to activate a mature, naive T cytotoxic cell? You have a minute. Go. Talk amongst yourselves, it's way too quiet in here. I know you guys are all recovering from Halloween and you're having that sugar overload this morning. Sugar overload. Okay. Ask Alec or Haley, they're here for you. Ask them if you're not certain. Of course, I haven't told you, asking you what the answer is. I'm going to hold you to the time limit on this one, people. Ten seconds. Oops. What happened? Okay. B7 binding to CD28 is the correct answer. That is the second. Yes, I know. People, again, I think people butt dial. Okay? So, let's go to the next one. So here is how a T cytotoxic cell, a T cytotoxic cell is going to be activated in a secondary lymphoid organ. A Peyer's patch, a salt, a mall. There's all sorts of things, okay? All of these things, lymph node. It's going to be activated there. It's going to migrate to the tissue via the selectins that are near where that inflammatory response is going. Because you're going to have inflammatory response. Because if you don't, you haven't talked about this. One of the other things that interferon alpha and beta does is to upregulate more MHC class 1 on the surface of the cell, which makes sense. You want to have more MHC class 1 on the surface of the cell to display more possible peptides to activate as many T cytotoxic cells to alert the immune response that you're infected, or that you could be. Am I normal? I'm normal. I'm infected. I'm normal. I'm normal. I'm normal. I'm infected. Okay? So then once the T cytotoxic cell has been activated in the paracortex, or in the paracortex, it's going to migrate, it's going to be drawn by chemokines out from the secondary lymphoid organs into the circulatory system, and it's just like a PMN, it's going to find those selectins that have been upregulated. If it's in the lungs, it'll be a velar in the capillaries near your lungs. If it's in your arm, it's going to be in the capillaries in the circulatory system near your arms. They're going to migrate out of your tissues and start killing off the cells that are expressing signal 1. They can't kill anything, it can't require a second signal to kill because how many lung cells have MHC class, have B7 on them? 0. 0 liver cells have B7 on them. 0 kidney cells have B7 on them. 0 muscle cells, 0 nervous cells, 0 anything but an antigen presenting cell. So we are going to selectively activate. We're going to have M activate James Bond. He's going to clonally expand. He's going to go off to find signal 1 and kill it. He's been shown what his target is by a dendritic cell. Look for this target and then kill it. And then they make a clone army and they do that. Does that make sense? For a T cytotoxic cell, they deliver a death package. You need to know the 2 names of the death package. The book talks about perforins. Perforins poke a hole in a cell. The enzymes that are also dropped in as a package, you can think of it as a nuclear bomb that's dropped into the house and they back away and explodes. Those are granzymes. So perforin and granzymes are the 2 effector molecules that T cytotoxic cells make to kill a cell. What do I mean by perforins and granzymes? I mean that, again, once an effector T cell or a memory T cell, because a memory T cell has received the second signal. It is a clone of that first activated T cell. The effector cells are clones of that first effector T cell, and all of them, because they're
clone of the original one have received the second signal because they're clones. So, this effector cell or memory cell only ever needs to be see signal 1 in order to kill. Once it sees that same peptide, it's licensed to kill that specific phase, and only that phase. Okay? This is a cancer cell, this is a T cell. These cancer cells, there's a macrophage that is eating a cancer cell that is activated, that has cross-presented a cancerous peptide. Cross-presentation means an antigen, a macrophage is eating a cancer cell, it chews it up, and it drops some of its proteins into its cytoplasm so that it can get into the MHC class 1 pathway. So now we can activate the T cytotoxic cell to these cancer cells. We have now activated the T cytotoxic cell. This T cytotoxic cell has recognized the MHC and the tumor peptide that's on this tumor cell. It delivers its perforins and granzymes, and we now have a dead tumor cell. We poke a hole, we drop a bomb, we back away, we kill another tumor cell. Until we are exhausted and we die, and we make more T cells until the tumor is gone. The book also we talked about M1 versus M2 cells earlier in the class, different kinds of macrophages. An M1 macrophage promotes inflammation, promotes the activation, promotes the killing of things like tumors. An M2 macrophage actually generates cytokines that stop the immune response. One of the things we have found in certain cancers is that in solid organ tumors, we find M2 macrophages in this cancer tumor, so that when that T cytotoxic cell comes to kill it, it sends out signals that say, these are not the drones you're looking for. Okay? Go away. These are not it. And it shuts down, that cytokine will bind to that killer T cell, and it will turn down. So this is the way tumors subvert the immune response. We see the opposite happen in people who have autoreactive autoimmunity. If you have juvenile diabetes in your pancreas, we need those M2 macrophages to shut down an inflammatory response, but instead what we see is an M1 macrophage response that promotes inflammation against self. We're trying to, as immunologists, switch those responses. We want M2 in autoimmune responses, we want M1 against tumors. We just don't know how to do it yet, and we don't know why tumors make M2 macrophages, how they generate those. We don't know what causes their formation. We just know that they're there. Okay? This is a T cytotoxic cell response. What happens when a memory T cytotoxic cell receives only signal 1? You have a minute. Talk amongst yourselves. Haley and Alec are there. Ask them questions. Go. She's there. She's willing to give you guys hints.
$15 seconds. Four, three, two, one. Whoops. It becomes fully activated, clonally expands, and produces memory cells. People are like, but you didn't say kill. It doesn't matter. That's what it does. That's one of the things it does. Any activated effector lymphocyte will do those things. Any of them.
except for B cell that only receives signal 1. If a B cell doesn't receive 2 signals, it dies. If a T cell doesn't receive 2 signals at the same time, it dies. So when we talk about becomes partially activated and starts to clonally expand, that's only B cells receiving signal 1. When we talk about migrates to a B cell for help, that would be an effector T helper cell. When we talk about migrates to a secondary lymphoid organ to find a professional APC to get full activation, I just made that up. That's the longer, more complicated answer that you guys always fall for on an exam. I keep telling you not to. Find the correct answer and pick it. Okay? It's deleted or energized. It's easy to pass my exam. You find the correct answer and pick it. It gets deleted or energized as if it only receives signal 1. Okay?
Okay, so now we're going to talk about how B cells get activated. How do we make an antibody response? What happens when a naive mature B cell receives only signal 1? Go. You have 30 seconds on this one. I told you what the answer was literally. Go.
I'm not being tricky here, people. Okay. 10 seconds.
Okay. The other thing is, before we go any further, I want to make sure that everybody has make sure that you have posted your blog posts to the assignment link, because if you don't, you're going to be marked late. Okay? As of today, as of from now on, you must get your blog post onto the homework, the assignments link, otherwise you're going to be marked late. Okay? Okay. The correct answer is, let's see how many people, come on people, nuts. Okay, but people say, well, but you didn't say then it dies from neglect. So I said, in the question, what happens when a mature naive B cell receives only signal 1? Okay, it's going to start, become partially activated, and clonally expand. Yes, it will eventually die from neglect, but that's what it's going to do. Okay? It is deleted or energized? No. It is not deleted, it dies from neglect. It has been partially activated. Not the same thing. T cells become deleted or energized. It is partially activated. It waits for a second signal, and if it doesn't get one, then it will die from neglect. Different from deletion or energy. Okay?
Also makes up IgM.
Absolutely perfect. It does also make some IgM while we're waiting. So the things that it will do, will become partially activated, it will clonally expand, it will migrate to the edge of the B cell follicle, and it's going to do all sorts of interesting things. Once it gets cross-linked, it's going to upregulate this. When a B cell gets cross-linked, when it migrates to the edge of the B cell follicle, it is technically an antigen-presenting cell, if it needs to be. It will upregulate MHC class 2, an pathogenic peptide. It will upregulate B7. It is going to want to get help from an activated effector T helper cell, so it's also going to upregulate CD40. Okay? All of those things by cross-linking 2 B cell receptors. So then, once we're going to finish off T cells, and we'll probably have to finish this off on Tuesday, but I'm going to try and get through some of this. When we talk about T helper cells, when they're naive mature, T helper cells are Th zeros. Once that antigen-presenting cell presents a peptide and its PRRs have been activated, it's going to tell it to become a Th1. The book talks about a Th1 being specifically for a cytolytic response. It's going to make 2 cytokines that you need to know. It's going to make IL-2, and it's not the only cell that makes IL-2, but it makes a lot of IL-2 to help T cytotoxic cells grow. That's what IL-2 does. It's also going to make specifically interferon gamma, because that's going to activate macrophages as well. We're also going to activate macrophages by binding CD40 and CD40 ligand on an activated macrophage. We can make a Th2 response. A Th2 response is specifically to help B cells become activated, and to make an eosinophil or an IgE response. The three cytokines that are involved, IL-4 is to help make antibodies. IL-5 and IL-13 are to activate eosinophils and make IgE. So when a Th2 goes to a B cell that has been partially activated, and it sends IL-5 and IL-13 to it, this B cell is going to class switch because it says that there's a parasite in the body and I need to make IgE. For Th17, we're going to create an inflammatory response that specifically activates, right, in big letters, PMNs. PMNs, okay? T follicular helper cells are the ones that are supposed to go in and help B cells. Okay, the follicular helper cells, the book talks about them not in the same place as all the other T helper cells, but the book talks about the follicular helper cell. And then when we talk about T regulatory cells were generated when a immature T helper cell bound to cell too strongly. If it binds to cell too strongly, the body says, it's binding to cell too strongly. We're going to use you to recognize the cell response and shut it off. So in that juvenile diabetes, not only do we need an M2 macrophage, but we need T regulatory cells to be present to shut down the inflammatory response by making IL-10 and TGF-beta. Those are the additional cytokines that you need to know besides IL-1, IL-6, and TNF. Are we good? Okay, so what else do we know? This is the overview of what happens. We have a dendritic cell migrate into the paracortex, going to activate a T helper cell, a Th0, be a signal 1, MHC class 2 in peptide, CD4, signal 2, B7, CD28. We're going to have a B cell that was cross-linked when that antigen came in, become clonally expanding, partially activated, migrate to the edge of the B cell follicle and call for help. You'll send cytokines out that draws activated T cells to you. So we're going to have them come out. We're going to have these short-lived extrafollicular T helper cells and when they're activated, they're going to upregulate CD40 ligand. They're going to activate CD40 ligand. They're going to activate the B cell, and they're also going to send whatever cytokines. If I send IL-5, IL-13, you're going to make IgE. If I'm going to make IL-2, you're going to make IgG. If I'm going to make Th17, you're going to make IgG because it's an extracellular bacterial infection. We're going to activate PMNs. There's all different flavors of IgG that you guys don't need to know about. Okay? This is from a different book, but I just wanted you to see what happens. And then there are going to be some activated T follicular helper cells that go into the follicle and help the B cells undergo clonal expansion, class switching, and affinity maturation. Okay? We're going to talk about this now. Once we get the B cell, if a B cell acts as an antigen-presenting cell, we just eliminate the dendritic cell and the T helper cell in the follicle. The B cell gets partially activated, becomes an antigen-presenting cell, upregulates B7, upregulates CD40, migrates here, processes MHC class 2 molecules, activates signal 1, signal 2 for Th0 cell. They check their T cell receptor against that. We find the correct Th0 cell that binds to her MHC class 2. The second signal is sent via B7. And then these T helper cells step back, start clonally expanding. And while they're clonally expanding, they're going to upregulate what on their surface? CD40 ligand. Then they come back to the B cell, and then they're going to bind CD40 ligand to their CD40. This is the second signal. You have to have CD40 ligand in order to activate class switching and affinity maturation. That signal is what activates a specific transcription step that allows the transcription and translation of the enzyme that allows that to happen. When that happens, We are going to have this happen. This is what we're going to have: the B cell, the T helper cell comes in, activates that B cell fully. The B cell will step back and it will clonally expand. Okay? When it undergoes clonal expansion, it's going to start class switching. We've already talked about how this works. We talked about how there are sections as these clones expand, they're going to actually go in, and that enzyme that we activated via CD40 ligand is going to cause DNA to be deleted. We are going to, just like you're deleting a program off your computer, we're going to delete the gene cassettes for IgM and IgD. Or IgM, IgD, IgG, and IgE if we have to make IgA. We're going to delete anything that the T helper cells told us to delete to make the correct antibody. Because our clones will never ever express IgM or IgD again, the B cell receptor on the memory B cell that has undergone activation that is a clone of the original cell will always have that B cell. If it's IgG, it's going to have a B cell receptor that's IgG, because that's the only heavy chain it knows how to make. If it's IgA, there'll be IgA as its B cell receptor. It will be a memory B cell, just by dint of having a different class of antibody on its surface, right? If it has IgM and IgD on it, what is it? It's a naive mature B cell. IgM and IgD, naive mature. When a naive mature B cell gets cross-linked, it will make IgM. It will migrate to the edge of the B cell follicle and link for CD40 ligand so that it can delete the DNA and make the appropriate heavy chain constant region. And then it's also going to undergo affinity maturation, where that same enzyme that deletes that entire section of DNA goes in and tweaks that VDJ section, or that V section, that V, the light chain variable region, or the heavy chain constant region, and it shrink wraps it so it binds absolutely, terrifically strongly to antigen. The same enzyme does both of those things. It class switches and it allows affinity maturation. And then, the other thing that I wanted to show you is this is a B cell that's been activated. Once it has been fully activated, it is no longer an antigen-presenting cell. It is now the job it's supposed to do. It is now a plasma cell to make antibodies. And generally, where we see the plasma cells that make antibodies are they migrate to the bone marrow, and they simply pump antibodies into the bloodstream, which is where the antibodies are supposed to go. If it's IgE, it gets into the tissues. If it's IgA, it's transferred to the mucosal. If it's IgG, it's where it's supposed to be. Does that make sense? Last question? We'll finish this up on Tuesday. Look over the rest of these slides